Yeah, good evening, everyone. I hope the audio, visuals, everything is fine, clear as of now for today. Yeah, good evening, Rimsha, Kanchandeep, Mohammed Saddam, Bas Badhiya Bhai, tum batao. Prikshit Siddhi, welcome to the session. Just give me a thumbs up in the chat box, guys, that the video, audio, everything is perfectly fine as of now for today. Yeah, Siddhi says, okay, so we are ready. Yeah, sure. Okay, perfect. Rimsha, perfect, perfect. Sure. Today we will be completing with the cell injury, the final shot of cell injury. So, yes, just waiting for another one more or two more minutes and let everyone join and then I'll start with the session. Yes. So, I hope you all are doing good at your ends. Sure. Let us start now. Yeah, it's already 8 as of now. Yeah. Mohammed Yasir, good evening. Welcome to the session. I'm doing very good. How are you, Yasir? Okay, sure. Now, today I'm going to discuss about gangrene first. And before coming to the term, just remember, guys, that gangrene is a clinical term. Gangrene is a clinical term. Okay, gangrene is a clinical term. Basically, we see and we realize that it's a gangrene. Okay, so gangrene is basically of two types: dry gangrene and the wet gangrene. So, first, we will be talking about the dry gangrene. First, we will be discussing about the dry gangrene. And remember, guys, what I'm trying to show you over here, that the blood supply to this particular area of the limb, for example, like foot or hand or any particular part of the limb, the blood supply is reduced. Okay, so we say that in dry gangrene, basically, it is the area of complete ischemia. Now, can anyone in the chat box tell me what do you understand by the term ischemia? I have already discussed it in my previous sections. So just let me know. Ritika, good evening. Nezi, good evening. Yeah, just let me know what do you understand by the term ischemia. And remember that if there is like, yeah, very good. Kanchandeep, it is the less blood supply or decrease in the blood supply. Okay, if suppose there is a decrease in the blood supply to a particular part of the limb, okay, and that is nothing which is seen in coagulative necrosis, that is seen in coagulative necrosis. Yes, Siddhi, less blood supply. Nisi, very good. Prikshit, very good beta, perfect. Perfect, guys. Yes, it is perfect. Yeah, it is the decreased blood supply or the less blood supply to a particular organ. Now, what will happen? See, whatever blood vessel is going to this particular organ, suppose this is an organ, okay, and this blood vessel is obstructed. So the amount of blood reaching to this organ will be decreased. Okay, the amount of blood reaching to this particular organ will be decreased. Now, what will happen? Uh, it will get less oxygen. That is very much fine. Along with that, remember, guys, that this particular organ will not be infiltrated. It will not be infiltrated with neutrophils. With neutrophils, remember, not infiltrated with neutrophils. Why? Because due to obstruction of blood supply, due to obstruction of blood supply. Osama Sheikh, good evening. Okay. Now the neutrophils won't be reaching these particular areas because there is an obstruction in the blood vessel. That is why there is like ischemia, less blood supply or decreased blood supply to that particular organ due to some obstruction. Now what will happen? Decomposition will take place. Decomposition will take place, and remember that decomposition will classically give a black color along with offensive smell, along with the offensive smell. So in dry gangrene, what you will notice a black color part of the limb along with a very offensive smell. You can see this black color, okay, and you will get a very offensive smell coming out of this. So that is all about the dry gangrene. I hope this is clear to each and everyone. It is seen in the coagulative necrosis. Okay. Yes. Just give me a thumbs up, guys, that you all have written this. You all have written this. Just give me a thumbs up, guys, in the chat box. Prikshit, fine, beta. Okay. Can anyone tell me in the chat box that what is the most common cause of coagulative necrosis? I have discussed it in the last class. What is the most common cause of coagulative necrosis? I'm waiting. The answer is already written on my presentation, but I want you people to answer this. Yes, Prikshit, perfect, beta. It is the ischemia. Yeah, that is the reduced blood supply to a particular cell or an organ. Yes, Dr. Y, it is ischemia. Perfect, beta. Perfect. I'm waiting for more answers, guys. You have to be like quick. जिस स्पीड से आप लोग टाइपिंग करते हो ना अपने रिस्पेक्टेड बॉयफ्रेंड्स और गर्लफ्रेंड्स को, उसी 64 kps की स्पीड से इसमें भी चैट बॉक्स में भी आंसर टाइप किया करो. मतलब आई वांट टू सी हाउ फास्ट, हाउ गुड यू आर एट नो रिप्लाइंग. इंटरेक्टिव सेशन बनाओ ना. ऐसे तो मैं तो लिखवा ही दूंगा सब कुछ, मैं तो बता ही रहा हूं, बट यू शुड लाइक पार्टिसिपेट. चलिए, कमिंग ऑन टू द नेक्स्ट पार्ट, दैट इज द वेट गैंग्रीन. Wet gangrene. I have already discussed that gangrene is a clinical term. We see and we classify, or we say it as gangrene. Okay, we diagnose it clinically. Okay, the very first one, that is the dry gangrene, I have already discussed. Coming on to the next part, that is the wet gangrene. Coming on to the next part, that is the wet gangrene. And remember that wet gangrene, it is basically dry gangrene plus superimposed bacterial infection. Okay, it is dry gangrene plus you will find a secondary infection over here that is called as the bacterial infection. Okay, that has just superimposed this gangrene. Okay, bacterial infection, superimposed bacterial infection, superimposed bacterial infection. Dry gangrene, it is basically the, in other words, I can say it is ischemia, that is the reduced blood supply. Okay, and apart from that, the superimposed bacterial infection, I can nearly say it as secondary infection. It is a secondary infection which is taking place. So it is wet gangrene is ischemia plus a secondary infection that has superimposed on it. It can be a bacterial infection as well. Wet gangrene is basically a type which is seen in liquefactive necrosis. Liquefactive, liquid और wet terms coincide कर रहे हैं ना, तो इस वजह से आप लोगों को याद रहेगा. It is liquefactive necrosis. It is liquefactive necrosis. Okay. Coagulative necrosis, it's dry gangrene And liquefactive necrosis, it is wet gangrene. It is wet gangrene. Sure. I hope you all have written and you all understood about the dry gangrene and the wet gangrene. It is all done. Coming on to the next part, that is apoptosis. Coming on to the next part, that is apoptosis. I hope everyone is participating, guys. And now this is a very important topic. From exam point of view, just put a dollar sign. Wherever I'm using dollar sign in my lectures, I already like it is indicated that this particular part is important, and you have to like, yeah, understand it. I hope today all the audio visuals are clear. Okay, Prikshit is fine. Okay, Yasir, good beta. Sure. Rimsha, Nazi, perfect. Sure. ठीक, बाकी लोग भी टाइप कर लिया करें. आई होप आज करवा चौथ तो नहीं है, तो सबके हाथों में मेहंदी तो नहीं लगी होगी. तो इट्स सिद्धि. Now guys, apoptosis is basically referred to as falling of leaves. Falling of leaves. You guys already know that just as the leaves get dried and no, the leaves get shed off, okay? Basically, in the similar pattern, the cell gets fragmented and the cell gets died, okay? In the similar pattern, just like leaves, the cell gets fragmented, and in a similar fashion, the cell dies. So it is referred to as programmed cell death. It is referred to as the programmed cell death. It is referred to as the programmed cell death. Okay. Now, why it is called as programmed cell death? Because it is controlled by set of genes. It is controlled by set of genes. It is controlled by set of genes. Okay. And that's why it is referred to as a programmed cell death in a series. Okay. Anything happening inside the cell, okay, it is just controlled by the genes. Okay. Yeah, it is just controlled by the genes. Apart from that, remember that apoptosis affects single cell or a small group of cells This is something which is important. Remember that it affects single cell or a small group of cells. It is dependent on the genes, okay, thereby referred as the programmed cell death, okay. And remember, yeah, it is dependent on which particular genes. Can anyone tell me? Okay, chalo, we'll discuss this. Yeah, we'll discuss this in another slide. Okay, we'll discuss this in another slide. So put the next heading as genes associated with apoptosis. Put the next heading as the genes associated with apoptosis. Now guys, there are three types of genes, majorly two, and the one which is balancing this particular process. The very first one, remember guys, it is the pro-apoptotic gene. Pro-apoptotic gene. Oh, pro-apoptotic genes. Then we have the anti-apoptotic genes. And the one which balances the both, it is the sensors. Remember guys, coming on to the pro-apoptotic gene. Pro, remember what from the word P, it promotes apoptosis. It promotes apoptosis. Anti-apoptotic genes, on the other side, it will inhibit apoptosis. And the sensors, remember guys, it will balance apoptosis. So we have three types of genes: the pro-apoptotic genes, which is promoting the process of apoptosis, the other genes which is inhibiting the process of apoptosis, and the third one which is trying to keep a balance between both the genes. Okay, yes, Ankita, very good. Nishant, good evening. Yeah, Madhavan, okay, very good, Madhavan. So now already you have mentioned it. I will try to mention it in my table as well. So the very first one, guys, yeah, whenever we are talking about the pro-apoptotic genes, we have write down the genes, that is the BAC gene, BAX gene. Then we have P53 gene, P53 gene, P53 gene. Then we have, okay, so these are the three genes which are promoting apoptosis. Okay, and out of this, just remember the third one. Just remember the third one, that is the P53 gene. It is the most important. It is the most important gene, okay, pro-apoptotic gene. Apart from that, there are certain drugs, guys, which are behaving like pro-apoptotic genes, or like they are promoting apoptosis. Can anyone tell me the name of that particular drug? That particular drug? Remember, these are glucocorticoids, 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 which are promoting the apoptosis. Yes, glucocorticoids, which are promoting the apoptosis. And apart from that, guys, remember, yeah, apart from that, guys, remember that this is the most important gene, that is the P53 gene, and the most important drug over here, it is the glucocorticoids that is promoting the apoptosis. Coming on to the next part, that is the anti-apoptotic gene. Anti-apoptotic gene. And in anti-apoptotic gene, remember, the most important anti-apoptotic gene, it is BCL2 gene. Okay, that is the most important one. BCL2 gene, that is the most important one. Apart from that, remember, we have BCLXL gene, MCL gene, MCL1 gene, okay, and the drugs which is in turn important, which inhibits the process of apoptosis. The drugs are sex steroids. Now, question can be in a very different way: which drugs behave like anti-apoptotic genes? It is the sex steroids. Or if the question says which drugs promotes apoptosis, it is glucocorticoids. Apart from that, remember which is the most important anti-apoptotic gene? It is the BCL2 gene. That is again a question. I'll put a dollar sign. Which is the most important pro-apoptotic gene? It is P53 gene. That again I'll put a dollar sign over here. Apart from that, in anti-apoptotic genes, if you guys want to remember, just remember the last letter. It is coming L. It is coming L. Okay, it is coming L in the last one. The last alphabet, that is the L. Yes. Apart from that, coming on to the sensors, which balances the apoptosis. So you will write it over here. We have BIM gene, BID gene, Puma gene, Noxa gene. Puma gene and Noxa gene. So this is a table, guys. I will try to like put a dollar sign. Remember some of the most important points over here. Okay, the most important ones are the most important. Okay, so yeah, apart from that, we will write some important one-liners in our notes. So just mention one point that BCL2 gene is associated with, it is associated with follicular lymphoma. It is associated with follicular lymphoma. Apart from that, the MCL1 gene, it is associated with development of resistance to chemotherapy. Chemotherapy. So guys, there are two different types of question that have been asked in the exams. If they simply say you, what is the most important anti-apoptotic gene, that is BCL2 gene. The other question is the most important anti-apoptotic gene which is associated with development of resistance to chemotherapy. It is MCL1 gene. So we have two different answers for two different questions. So these 2 points are, yeah. So these 2 points are anyway important. Just put a dollar sign. These are one-liner questions. And apart from that, remember that one more anti-apoptotic gene. Okay, not anti-apoptotic, the pro-apoptotic gene. It is BCL-XS gene. Okay, BCL-XS gene. Now over here L is coming, so don't confuse it and write it over here. It is wrong. It comes in the category of pro-apoptotic gene. It is the only differentiation that I want to tell you. Okay, so it is the BCL-XS gene which comes in the category of pro-apoptotic genes. Although I have already discussed that the genes ending with L comes in anti-apoptotic. This one is quite difference that you guys have to learn. So I hope this is clear to each and everyone, guys. Just give me a thumbs up so that I can start with the next part. That is the difference between the apoptosis and the necrosis. Just give me a thumbs up in the chat box, guys, that you all have written and understood the genes associated with apoptosis. Yes, Prikshit, Siddhi, perfect. Omkar, Gaikwad, बच्चे perfect, perfect बेटा. Waiting for more thumbs up, guys. Yeah, sure, perfect. Coming on to the next topic, that is the difference between necrosis and apoptosis. Difference between necrosis and apoptosis. Yeah. So yes, Shubham Rao, Biomed, Funkies, Kanchandeep, Muskan, Madhvan, Ruben. Okay, perfect, perfect, guys. Please participate in the class as much as you can. Okay. Now, guys, there are certain features, and on the basis of these features, I will try to differentiate between necrosis and apoptosis. Now, guys, coming on to the very first one, that is the definition. Definition. That is the definition, guys. That is the definition. Now, remember that what is the definition of necrosis? You already know, I have discussed. It is an enzymatic process. Okay, enzymatic or ischemic process. Ischemic process. And when we talk about apoptosis, apoptosis process is depending on genes. It is depending on genes. We have already studied that there are pro-apoptotic genes, anti-apoptotic genes, and the sensors. So it is a programmed cell death. Okay, it is a programmed cell death and it is dependent on genes. So I can write it as genetically programmed cell death. Programmed cell death. Genetically programmed cell death. Genetically programmed cell death. Coming on to the next feature. Coming on to the next feature, process. Process. Remember that necrosis is basically a passive process. Now, guys, can anyone tell me when I say passive, what is the definition of passive in medicine? What is the definition of passive in medicine? Can anyone tell me in the chat box from their physiology knowledge? I hope you all are like masters in physiology, I suppose. Hmm. I'm waiting for the answers. Yes, Kanchandeep, very good. It is without energy. Necrosis is taking place without energy, whereas when I compare necrosis with apoptosis, it is taking place with the help of ATP. Yes, Sakshi Kumari, very good, beta. Yeah, it is without no ATP is required. Whereas when we talk about apoptosis, it is it is an energy dependent process, or I can simply say it is an active process. It is an active process. Yes, Osama, Prikshit, perfect. Coming on to the cell death. The cell death. Remember, a large group of cells is involved in necrosis. Okay, it is basically a large group of cells. Yes, Omkar, Siddhi, very good. Large group of cells. Apart from that, in apoptosis, I have already told you it involves a single cell or small group of cells. It involves a single cell or small group of cells. Is it lagging? Coming on to the types. The types. We have two types of necrosis. We have only one type of necrosis, that is always pathological. Pathological. It is associated with disease, whereas apoptosis is seen both physiological and pathological. Physiological and pathological. Guys, just let me know in the chat box if everything is clear, audio, visuals, everything is fine or not. Yeah. Is it like not working as well? Coming on to the next part, that is the cell size. Now, can anyone tell me what happens to the cell size in necrosis? Can anyone tell me what happens to the cell size in necrosis? Can anyone tell me what happens to the cell size in necrosis? Okay, it increases or decreases? Yes. The cell size in necrosis, Mohammed, it is cell size. I'm talking about necrosis right now. No. The cell size increases. Whereas when we talk about apoptosis, it decreases. It decreases in apoptosis, whereas the cell size increases in necrosis. Yes, Kanchandeep. Yes, yes. Apart from that, remember the next feature. What happens to the cell membrane? That is again a very important question. I'm putting $2 signs over here. Can anyone tell me what happens to the cell membrane in case of necrosis? Yeah, the cell membrane, there is an activation of an enzyme called as phospholipase. And you already know that the cell membrane, it is composed of phospholipids. The cell membrane, it is composed of phospholipids. So yeah. So what will happen to the cell membrane over here? It will get damaged. Yes, it will get damaged. Whereas when we come to the next part, that is apoptosis. Since there is no activation of phospholipase guys over here, the cell membrane is intact. The cell membrane, it is intact. The cell membrane, it is intact. There is no activation of phospholipase in apoptosis. We'll just discuss the pathogenesis in another few minutes. But before that, just complete this table. Coming on to the next part, that is the inflammation. Another feature on which we can distinguish between the necrosis and the apoptosis. The inflammation, it is present in case of necrosis, whereas it is absent in case of apoptosis. Okay, it is absent in case of apoptosis. Talking about the next feature, that is the marker. That is the marker. And remember, we have no marker for necrosis, whereas coming on to this, we have annexine, which we can use to detect, okay, CD95. So these are certain markers of apoptosis, okay. And on gel electrophoresis, that I will discuss in a couple of minutes, gel electrophoresis, you will see a smear pattern, okay. And over here in apoptosis, you will see a step ladder pattern. You will see a step ladder pattern. You will see a step ladder pattern. So gel electrophoresis wala point I will be discussing in another few minutes. But before that, just remember we have just combined, or we have just compiled all the differential points in one particular slide. Okay, so that is again a very important slide that you can mark with a dollar sign and just try it to remember like before the exams. Okay, now coming on to the next part. I hope everyone has written up till here. I hope everyone has written up till here. Just give me a thumbs up. Yes. Perfect. I'm waiting for your thumbs up, guys. I hope you all have written it. Is it some way like okay? So far now, Prikshit, Siddhi, okay. Perfect, beta. For now, we have discussed about the differences between necrosis and apoptosis. We have discussed about the genes that are associated with apoptosis. We have discussed about the types of gangrene. Now coming on to the next part in apoptosis, it is the physiological apoptosis. It is the physiological apoptosis. Now can anyone tell me physiological apoptosis? It is basically seen in which conditions? Write down the very first one. It is the embryogenesis. Embryogenesis. Now, when we talk about embryogenesis, how physiological means it is natural, it is likely to happen. Okay. Now, coming on to in embryogenesis, how for development of the upper limb. Suppose for I'm talking about the development of this particular limb. I'm talking about the development of this particular limb. Now, in case of hands, if all the cells between the digits, now hand is like this, so we cannot use it until and unless the cells between these fingers, between these digits die. So this takes place by the help of apoptosis. The cells in between the digits die, and you can like the fingers are separated, and you can use your finger. Okay, so that is simply at the time of embryogenesis. Okay, so physiological apoptosis is seen. So all the cells in between the digits are separated. Okay, now if suppose guys the cells don't separate, so what is this condition called? What is this condition called? Just tell me in the chat box. I'm waiting for your answers. I just want to test your knowledge. Yes, Osama Sheikh, it is syndactyly. Very good, beta. It is syndactyly. Very good. Very good. Perfect, beta. Apart from that, if we come, yeah, the menses in female. Okay. The menstrual cycle. Yeah, it is syndactyly. Rimsha, codes of literacy. Very good. It is syndactyly. Yeah. Apart from that, in menstrual cycle, guys, the females of the reproductive age group. Okay. Females of the reproductive age group, you already know due to the hormones, okay, the estrogen and the progesterone, the endometrium layer, okay, like it is formed, and as soon as the levels of progesterone decreases, what happens? The endometrial lining is shed off. Okay, that is why the menses are taking place. So over here, the endometrial cells also die. So that is again an example of physiological apoptosis. Apart from that, guys, remember the third one and the most important example, that is the apoptosis of increased TLC. That is the total leukocyte count. That is the total leukocyte count. Can anyone in the chat box tell me what is the normal value of the total WBCs present in human blood? What is the normal value of total WBCs present in the human blood? Yeah. So apoptosis of increased TLC after infection subsides. Now I'll explain the point. Infection subsides. Now suppose guys, if there is any particular infection, okay, in the blood due to bacteria, due to virus, if there is any infection in the blood, what will happen? The leukocyte count, okay, the normal leukocyte count will increase, okay, because of infection. More cells will be produced to fight from that particular infection. Okay. Yes, Osama Sheikh, it is very much right. Yes. Yes, Osama Sheikh, it is very much right. Now, what happens as soon as the infection subsides? What will happen to the increased TLC in the body? We don't need that TLC. We don't need that increased number of WBCs. So what will happen to them? They will undergo apoptosis. So that is again a very natural process. That is why it is referred to as the physiological apoptosis. That is why it is referred to as the physiological apoptosis. Okay? Yeah. Apart from that, one more example that I wanted to discuss with you guys, it is self-reactive B and T cells. Self-reactive B and T cells. Yeah, Osama, it is 4K to 11K. Yeah, very good. Yeah, it is the normal, and in certain cases, certain infection, guys, certain infection, basically the WBC's number is increased, TLC is increased. आप blood test करवा के देखोगे patient का. If you do the blood test of the patient, you will find that the number of TLC is very much higher than the normal values. And as soon as the infection subsides after a few days, the increased TLC, okay, that will undergo apoptosis, and that is again a very natural process. Coming on to the fourth type, that is the self-reactive B and T cells. Now, can anyone tell me one thing in the chat box? B and T cells are produced from? They are the cells of what? चलो, I'm waiting for your answers. देखते हैं तुम लोग को कुछ पता है. B और T cells अल्फाबेट वाले cells नहीं है. WBCs, B cells, T cells, bone marrow, thymus. Yeah, bone marrow and the thymus. Yes. Lymphocytes. Now what happens, guys? There are certain B and T cells. No, if due to certain mutations or something like that, if B and T cells are like, okay, we have much more answers in the chat box. Yeah, Kanchandeep, yeah, it's right, Farma, yeah. If right, perfect. Now what is happening if there is a B and T cell which is like infecting our own body, okay, which is like harmful to our own body, okay? So these B and T cells are further destroyed. They are further destroyed by the process of apoptosis, okay? They are not needed by our body because they are reacting to our own immune system. They are like harming our own immune system, and in that case, like the self-reactive B and T cells undergo apoptosis, okay, which is these are abnormal B and T cells which is like harming our own body, affecting our own immune system. So in that case, the physiological apoptosis takes place. I hope this is clear to each and everyone over here. The physiological apoptosis, the examples I have discussed it. Just give me a thumbs up in the chat box, guys, that you all are done with it. Yeah, just give me a thumbs up in the chat box that you all are done with it. Yeah, autoimmune conditions. Yeah, very good codes of literacy. Prikshit, Kanchandeep, codes of literacy. Perfect, beta. Yes. Madhavan, I'll just start with the discussion on the pathways now. Yes. Perfect. 5G, knowledge of education and motivation. Such names are so... तुम लोग का दिमाग जितना कॉम्पिटिटिव है ना, मतलब इन चीजों को लेके कि हाउ यू गैदर अप सच आइडियाज, तुम लोग को तो सबको ऑस्कर मिलना चाहिए भाई. Coming on to the next type, that is the pathological apoptosis. Now guys, you already know in a cell we have the nucleus, and in the nucleus we have the DNA. If suppose there is any damage to the DNA, due to ultraviolet rays, radiation, chemical factors, biological factors, physical factors, if there is any damage to the DNA of the cell, now will the cell survive? Now DNA is the controller of the cell. If the DNA dies, okay, so ultimately it will lead to apoptosis. The cell will not survive. So any DNA damage comes in the category of pathological apoptosis. Any DNA damage comes in the category of pathological apoptosis. Any DNA damage comes in the category of pathological apoptosis. Okay. Yeah, Kanchandeep, Prikshit, yes, it is right. Apart from that, coming on to the next example, okay, that is the duct obstruction. Duct obstruction. Now you know that we have several ducts in salivary glands. Watson's duct is there. In pancreas, hepatobiliary duct is there. Now if there is any duct obstruction, okay, due to atrophy of the cells, so ultimately what will happen? The pathological apoptosis. Okay. Apart from that, guys, remember we have viral infection of hepatocytes. Okay, that is councilman bodies. Councilman bodies. That is typically referred as the apoptotic bodies. Now I will show you this with the help of a microscopic picture I have included over here. So this is basically seen in cases of viral hepatitis. Viral hepatitis. I've just taken one picture, microscopic picture. In viral hepatitis, you will be able to see this particular structure that is called as councilman bodies. Councilman bodies. Now what you see over here, remember this red color, which I'm just trying to show, it is denoting chromatin condensation. Chromatin condensation. Okay, the chromatin is condensed. Okay, you can see a chromatin is condensed, and apart from that, you can see the eosinophil. Here, it is much more darker than the others. So remember that it is hyper. The cytoplasm, I'm talking about the cytoplasm, bache. You will see the hyper eosinophilic cytoplasm. Hyper eosinophilic cytoplasm. Dark pinkish color you will be able to identify just near the nucleus, this hole. Okay, that is basically referred to as like more eosinophils are accumulated in the cytoplasm. Hyper eosinophilic cytoplasm with chromatin condensation gives the classical appearance that is called as councilman bodies. That is called as councilman bodies, and that is something which is referred like it is seen in viral hepatitis, or I can see these are the signs of cell damage or cell death. Okay, whenever you see this councilman bodies, it means that it is showing you a condition which is referred to as pathological apoptosis or cell death over here. Okay, if everyone understood, guys, this, just give me a thumbs up that you all understood. These councilman bodies are apoptotic bodies, beta. Apoptotic bodies, and if these are present, it indicates the cell has died. It indicates the cell has undergone death. Yes. It indicates the cell has undergone death. Yes. Perfect. Perfect, beta. विमल, उसामा, प्रेक्षित, रिमशा, बाकी सबके हाथों में तो आज कुछ हुआ है लगता है. ज्यादा हाथ इस्तेमाल करने से भी कमजोरी आ जाती है, ऐसा मैंने कहीं लिखा हुआ पढ़ा था, तो इसलिए तुम लोग नोट्स नहीं लिख रहे हो ना. Guys, you have to be like fast. केबीसी में होता है ना, मतलब फास्टेस्ट फास्ट. चलो guys, coming on to the next example, guys. Coming on to the next example. प्रेक्षित is laughing. प्रेक्षित, मेरा जोक बहुत ही लाइक पॉजिटिव मैनर में था. जैसे आप लोग लिखते वक्त आप लोगों का हाथ दर्द हो जाता है ना, इसको अपनी मानसिक स्थिति के हिसाब से एडजस्ट ना करें. Priyanshi, Sakshi, welcome to the session. Like, I don't know. Coming on to the fourth example, or the pathological apoptosis. We are discussing pathological apoptosis. Yeah, we are discussing pathological apoptosis. And the fourth one, it is the transplant rejection, guys. It is transplant rejection, also called as the graft versus. Okay, we have two different terms over here: graft versus host disease. Graft versus host disease. Now, guys, transplant rejection. For example, let us take the example of a patient who is undergoing a kidney transplant. So we are taking the kidney of the donor. Okay, we are taking the kidney of the donor, and this is the recipient. We are transferring this kidney to this particular recipient. Now, the body of the recipient or any other body, अगर कोई भी foreign particle, if any foreign agent tries to enter your body, what will the body do? Body will try to fight it. Okay, so it will again not allow this particular kidney to survive in its body, and that is nothing other than the transplant rejection, where you will find the pathological apoptosis taking place. Apart from that, what happens? In graft versus host disease, what will happen? Now, suppose if this particular kidney goes into the recipient body and this kidney starts like infecting the body of the patient. Earlier, the recipient body was like not allowing this kidney to stay over there. But now what is happening? This particular kidney is producing a negative effect on the body's immune system. So that is graft versus host disease. So this is dominant in this case. So in turn, I'm just trying to say that whether it is a transplant rejection, the organ is rejected by the host body, or whether we have transplanted an organ that is a threat to the host body, that's not allowing or that's not matching, in that case, basically apoptosis will take place, and that is nearly, it is a pathological apoptosis. Pathological apoptosis. Okay. Apart from that, one more term that I wanted to discuss with you people. One more term that I wanted to discuss with you people. The fifth one, that is the retinitis pigmentosa. Retinitis pigmentosa. Can anyone tell me what do you understand by the term retinitis? Can anyone tell me what do you understand by the term retinitis? आई विमल, आई तो मुझे भी पता चल रहा है, रेटिना से तुम लोग आई बोल रहे हो। आइटिस जहां पे भी लग जाता है ना बेटा, इन्फ्लेमेशन हो जाती है ना, रेटिना की इन्फ्लेमेशन रेटिनाइटिस हो गई। चलो ये तो बहुत अच्छी बात तुम लोगों को समझ में आ गई। You know, inflammation in the retina. Very good, Sakshi. Yeah. Now, can anyone tell me which two cells are found in the retina, or which are the principal cells of retina? In the chat box, guys. Kanchandeep, yes, it is right. Sakshi, very good, beta. Prakshit, very good, beta. Can anyone tell me the two major cells of the retina? I'm waiting for your answers. Yeah, rods and cones. Vimal, very good, beta. Yeah, it is the rods and the cones. It is the rods and the cones. Yes. Okay. Rods are responsible for raat wala vision. Okay, or I can say dim light vision. और जिसको हम लोग क्या बोलते हैं, रात्रि। रॉड से रात याद रखना ना। अंधेरे में रॉड सेल्स आर फंक्शनल, और कोन्स सी से कलर। So basically it is responsible for daylight vision. Yes. Scotopic और photopic vision. ठीक है विमल, I also agree with the statement. Dim light हो गई या रात वाली vision हो गई, अंधेरे वाली vision रात्रि के समय rods, और daylight vision it is the cones. Now guys, remember that it is the condition where there is, write down with a red marker, I'm linking pathology with ophthalmology right now, where you can see the excessive apoptosis of rods. If it is present, then it will lead to night blindness, okay, which is nothing but retinitis pigmentosa. रात में दिखना बंद हो जाएगा, अंधेरे में, रात में तो नहीं है। Night blindness, it is simply referred to as the night blindness, guys. It is simply referred to as the night blindness, guys. So that is all about if there is excessive apoptosis of rods. अगर rods की apoptosis हो गई, rods की cells जाती जा रही हैं, which is responsible for seeing in the dim light, so ultimately you won't be able to see in the dim light. Ultimately what will happen? It will lead to night blindness. That is nothing but the retinitis pigmentosa. So these are some cases of pathological apoptosis. These 5 cases I have already discussed with a microscopic image, which is very much important. Hypereosinophilia, chromatin condensation, that is seen in case of viral hepatitis, that is the Councilman bodies, that is the apoptotic bodies. So pathological apoptosis done, physiological apoptosis done, differences between necrosis and apoptosis done, the genes associated with apoptosis done, apoptosis concept, and the gangrene. So just give me a thumbs up, guys, that you all are with me and have written all the notes up till now. And then I will be discussing a very important topic right here in front of you, that is pathways of apoptosis. Just give me a thumbs up, guys, that you all have written, understood, learned. Yes. Prakshit, Siddhi, Vimal, Kanchandeep, Osama, Sakshi, Priyanshi, very good beta, very good. आप साथ साथ नोट्स लिखते रहिए, याद करते रहिए. आई गारंटी यू नोट से बाहर नहीं आएगा कोई भी क्वेश्चन. यू जस्ट हैव टू बिलीव मी एंड अंडरस्टैंड द कंसेप्ट. Just have to believe me and understand the concept. Chalo. Now guys, we will start with the pathways of apoptosis, and remember we have two pathways of apoptosis: the intrinsic pathway, also called as the mitochondrial pathway. सर बिलीव है तभी लेक्चर्स अटेंड करते हैं. माइटोकॉन्ड्रियल पाथवे, थैंक यू. इंट्रिंसिक पाथवे को माइटोकॉन्ड्रियल पाथवे भी बोलते हैं. व्हाई? बिकॉज इट इज टेकिंग प्लेस इनसाइड द माइटोकॉन्ड्रिया. आई जस्ट टेल यू हाउ. And the other pathway of apoptosis that we are going to discuss today, it is extrinsic pathway. It is extrinsic pathway. Now guys, we will first start with the intrinsic pathway. Intrinsic pathway, also called as the mitochondrial pathway. Also called as the mitochondrial pathway. विमल अभी समझ में आ जाएगा. जस्ट बी विद मी एंड ट्राई टू अंडरस्टैंड द कंसेप्ट विद मी. ओके, चलो. अब आपको पता है कि सपोज पहले हम लोग इंट्रिंसिक पाथवे पढ़ेंगे. सो जस्ट गिव द हेडिंग एज इंट्रिंसिक पाथवे. ओके, जस्ट गिव द हेडिंग एज इंट्रिंसिक पाथवे. Okay, or commonly called as the mitochondrial pathway. So we'll just start with the intrinsic or the mitochondrial pathway. Okay, and now we are going to learn what is happening over here. So first I will just try to draw a cell, and you already know a cell basically contains. Now suppose this is a cell, हमारा प्यारा cell, and a cell contains a structure that is called as mitochondria. So I will draw a mitochondria inside. Now can anyone tell me in the chat box mitochondria में कितनी layers होती हैं? How many layers are there present in mitochondria? I just want to test your 11th class knowledge. You have or not? My 10-gram peanut size brains, please make it 100 gram and just let me know in the chat box ki how many layers of mitochondria you know. Yeah, Vimal, it is 2. Kanchandeep, Priyanshi, wah, bachche wah. I'm like, very good. We have 2 double layered mitochondrial structure that we are going to draw right now. And inner structure, you already know their presence of crista. I have already discussed in my last session. The ones who didn't watch the video, kindly go through that video as well. Okay. Now this is the structure of a mitochondria that is found inside the cell. That is found inside a cell. Okay. Yes. Osama, Rimsha, Yasir, Prikshit, perfect. That is found inside a cell. Now guys, remember, you already know that mitochondria contains cytochrome C. Now there is presence of cytochrome C inside the mitochondria. Okay. Normally, a cell contains mitochondria, and in the mitochondria, you will find the cytochrome C. Now, this cytochrome C doesn't comes out in the cell. Okay. Cytochrome C cannot come out of the cell. Why? Because there is some structure which is preventing. Okay. There is some genes which is preventing this particular structure to come out of the mitochondria into the cytoplasm of the cell. Mitochondria के बाहर क्या है? पूरा का पूरा cytoplasm है, पूरा का पूरा cytoplasm है. So these structures are something which is known as the gatekeeper of the mitochondria, and this is something called as the BCL2 genes. Now they don't allow the cytochrome C to come out. They don't allow the cytochrome C to come out. ये आने नहीं देते. Now you already know, if suppose any watchman it is working in your society, he will only work when you will give him salary. Just when you stop the salary, the watchman will not work. Similarly, this BCL2 is also dependent on certain growth factors. It is dependent on certain growth factors. If present, BCL2 will function normally. If the growth factors decrease, the BCL2 will stop functioning. The BCL2 will stop functioning. Till now, is everything clear? Just give, let me know in the chat box. Till now, is everything clear, guys? Then I will draw a flow chart for you people. Kanchandeep says yes. Yes, Siddhi. Very good. Osama. Okay. Now, guys, just try to write it in the flow chart. Yeah, Prikshit. Good. Good. Just try to write it in the flow chart, guys. Ha. Yes, Vimal, you are very much right that cytochrome C helps in oxidative phosphorylation as per your biochemistry knowledge. It helps in the production of ATP. Yes, I agree with the fact. Now I'll discuss what is actually happening over here. Now suppose if there is a decrease in the growth factors. Now suppose if there is a decrease in the growth factors, what will happen? It will lead to decrease in the BCL2 activation. BCL2 will not work properly, or it will not act, it will not work properly. Okay, usko salary nahi doge, it will not work properly. There is a decrease in the function of BCL2. And what is the function of BCL2? It will not allow the cytochrome C to pass into the cytoplasm. बाहर क्या है? साइटोप्लाज्म है. माइटोकॉन्ड्रिया के बाहर क्या है? साइटोप्लाज्म है. ये सेल मेम्ब्रेन है. ये सब चीजें तो आप लोगों को पता ही है. ये सब चीजें तो आप लोगों को पता ही है. Now guys, remember that what will happen if this BCL2 is not functioning properly? Okay, what will happen? Another gene, another gatekeeper will start functioning, and I'll just draw it with another pen so that you guys like don't merge up. So remember that another gatekeeper will start functioning in place of BCL2, and that is something which is referred as BAC or BACs. Now, can anyone tell me BAC or BACs? Right now we have written which type of apoptotic genes are they? Which type of apoptotic genes are they? BAC or BACs? Now what will happen? This decrease in BCL2 activation because of decrease in the growth factors, what will happen? It will be replaced by, yes, replaced by, everyone is saying right, Osama, Priyanshi, Shakshi, Prikshit, yeah, Kanchandeep, it is pro-apoptotic. Now BCL2 replaced by back or BAX gene, back or BAX gene. Yes. Now what will happen? They will allow the cytochrome C to pass through this and come into the cytoplasm. Okay. Now they will increase the mitochondrial permeability. Mitochondrial permeability. And what will happen? This cytochrome C will leak into the cytoplasm. Now this cytochrome C will come into the cytoplasm. So there will be increase cytochrome C in the cytoplasm. There will be increased cytoplasm C in the cytoplasm. Now, what will happen? Cytochrome C, as Wimmel told that it is used for oxidative phosphorylation, it is no more there in the mitochondria. So the structures are automatically destroyed. Now, what will happen after this? I will just write it on the next slide. I'll just write it on the next slide. Just give me a thumbs up, guys, that till now you have understood each and everything. Whatever I'm just trying to explain you the mechanism, just give me a thumbs up in the chat box. Till now you understood each and everything. Chalo, Prikshit, perfect, perfect beta. Osama, perfect, perfect beta. चलो आगे लिखेंगे बेटा, उसी फ्लो चार्ट को कंटिन्यू करना है. तो आई एम जस्ट, तो वहां पे क्या हो गया था? इंक्रीज साइटोक्रोम सी इन द साइटोप्लाज्म. देखो बच्चे, कंचनदीप, BCL2 ने फंक्शन करना बंद कर दिया. BCL2 का काम क्या था? साइटोक्रोम सी को साइटोप्लाज्म में आने से रोकना. BCL2 was acting like a gatekeeper to stop the cytochrome C leakage into the cytoplasm. Growth factors की कमी की वजह से BCL2 also started like dysfunction of BCL2, or there is a decrease in the number of BCL2. Ultimately, BCL2 decreases. What will happen? The other apoptotic genes, that is BAC and BAX, will start functioning more. Okay, there's a balance now. One start decreasing, the other one will increase in number. The BAC and BAX will function, and the BAC and BAX are basically serving, or they are increasing the mitochondrial permeability. They will allow the cytochrome C to come into the cytoplasm. They will allow the cytochrome C to leak into the cytoplasm. They are paving the way for cytochrome C to come out, which was inhibited by BCL2. BCL2 inhibit कर रहा था, बैक और बैक्स उसको प्रमोट कर रहा है. बैक और बैक्स क्यों बढ़ गए? क्योंकि BCL2 का काम कम हो गया, तो बैक एंड बैक्स ultimately took up their function. Okay, so that's why. I hope it's clear as of now. Kanchandeep, I hope it's clear as of now. Yeah. Now after that, after that, what will happen? That there will be increased activation. Okay, sir, now clear. Okay, beta, perfect. Increased activation of APAF1. Now, can anyone tell me what is the full form of APAF1? Can anyone tell me what is the full form of APAF1? It is related with apoptosis only. It is related with apoptosis only. Yes, it is apoptotic protease activating factor. Apoptotic protease activating factor. Apoptotic protease activating factor. Apart from that, there is increased activation of apoptotic protease activating factor due to more accumulation of cytochrome C in the cytoplasm. It will lead to an increase in the activation of caspase 9. Very, very important. Caspase 9. It will lead to an increase in the activation of caspase 9. Now, you guys should remember that apoptosis is something which is dependent on caspases. Now, what are these caspases? Caspase are, so we'll just mention about some points regarding caspase and then I'll return back to the slide. So just leave 4-5 lines and just write a term that is caspase. So caspase is nothing but cysteine containing, okay, protease. It is containing cysteine protease acting at aspartic acid residues. Aspartic acid residues. Now you already know a protein made up of amino acids. Amino acids, this is aspartic acid. So it is basically an enzyme. It is an enzyme which will cut or which will break the protein structures where it will find the aspartic acid. Where it will find the aspartic acid, it will try to destroy that particular protein. And you all know that proteins are the structural units of the cell. If the structural units of the cell are destroyed, it ultimately means that the cell will undergo apoptosis, the cell will die. Okay, so it is cysteine containing protease basically acting at aspartic acid residues. They are destroying the proteins of the cell, the structural part of the cells, and the caspases basically numbered from 1 to 10. You can see over here the caspase 9, which is functioning in intrinsic pathway. Now, how to remember, guys? You know, 9 you have to write like this, and remember 9 may in RI, that is something that is intrinsic pathway, intrinsic pathway, intrinsic pathway. Now it is the yeah, it is the initiator caspase. It is the initiator caspase, and after that it will stimulate caspases 3, 6 and 7, which will lead to increased activation of proteases and endonucleases, which will lead to increased activation of proteases and endonucleases. देखो प्रोटीएज प्रोटीन को डिस्ट्रॉय करने वाला, endonuclease something which is endonucleases. Guys, can anyone tell me endonucleases is destroying which structure? We've already discussed about the chromatins. Okay, so ultimately the nucleus contains DNA, and DNA basically cuts the DNA at specific sites. So endonucleases, it's something which is destroying the DNA, and ultimately if the proteins and the DNAs, the endonucleases, the chromatins are destroyed, it will lead to cell death. It will lead to cell death. So remember that increasing activation of both these will lead to cell death. Okay, so remember that. Just write down 2 more points over here, that in intrinsic pathway, also called as the mitochondrial pathway, because it involves the functional mitochondria. What is the initiator caspase, guys? Can anyone tell me the initiator caspase involved? And what is the executioner caspase that is involved? Executioner caspases which is involved. Just from the flowchart, if anyone can answer these two questions which have been asked in the exams many times. Osama Sheikh 9, yes, it is the initiator caspase. Yes, Osama, perfect beta. It is the 9. I already told you, just write like this, you will remember initiator. Okay, intrinsic pathway. Initiator, intrinsic pathway. And what about the executioner pathway, guys? Anyone? Anyone would like to use his 5-gram peanut size brain? Please, guys. Yeah, coming after the caspase 9, that is the executioner caspase, that is 3, 6, and 7. Yeah, that is the 3, 6, and 7. So, yes, that is about the intrinsic pathway, guys, where I have already told you that it is the mitochondrial cytochrome C which is leaking into the cytoplasm. Cytochrome C, it is important for the oxidative phosphorylation in mitochondria for the generation of ATP and other process. But if cytochrome C leaves the mitochondria, ultimately what will happen? It will like lead to activation of certain caspases, and ultimately certain enzymes will be activated, which will lead to cell death. Which will lead to cell death. Yes, Prikshit, perfectly fine. I hope, guys, right now it is clear to each and everyone the intrinsic pathway mechanism. Just give me a thumbs up. Just give me a thumbs up in the chat box that the intrinsic pathway mechanism is clear to each and everyone. Rimsha, yes, perfect beta. What about rest, guys? Please, what about rest, guys? बाकी सब भी थोड़ा बहुत आंसर दे दिया करो, थोड़ा मुझे भी लगेगा कि नो आई एम टीचिंग सम गुड पीपल. या चलो, आयमन अहमद, प्रियांशी, प्रीक्षित, परफेक्ट परफेक्ट बेटा. Sure. Now coming on to the next, that is the mechanism of extrinsic pathway. Now coming on to the next, that is the extrinsic pathway. And over here, I will be discussing the mechanism of the extrinsic pathway. The mechanism of the extrinsic pathway. First, I will just try to illustrate with the help of a diagram. Now, guys, remember that suppose this is a cell. This is the structure of a cell. Okay, this is the structure of a cell. Okay, and this is something which is called as the plasma membrane. Okay, this is something which is called as the plasma membrane, or commonly called as the cell membrane. Commonly called as the cell membrane. Commonly called as the cell membrane. Now, guys, remember that like in the cell membrane, okay, on the cell membrane there are certain receptors. There are certain receptors, okay, which is commonly called as, or I'll just write it with another pen so that you guys are like more. So these receptors are T. Okay, I'll just write it over here. So these receptors are something which is known by the name of TNF. Okay, TNF receptors. Okay, also called as the FAS receptors. Okay, also called as FAS is also called as the CD95. Okay, in either ways. Okay, so these are certain receptors which are present on these plasma membrane or the cell membrane of the cell. Okay, and these receptors, you already know what is the function of the receptor: to perceive something, hormones or other chemicals or something, 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 something. So what happens? There is something which comes and bind to these receptors, and this is which is called as the Fas ligand. Now again, clearing the term that Fas receptor is this thing. Fas ligand is something which will come and bind to this Fas receptor. Okay, ligand is something which will come and bind to this Fas receptor. Okay, so Fas ligand or TNF alpha. TNF alpha will come and bind to TNF receptor. So we have the receptor, and to this receptor there are certain components which will come and bind. It is Fas ligand along with TNF alpha. Along with TNF alpha. Now what will happen? As soon as these ligands and TNF alpha bind to these receptors, what will happen? What will happen? There is a structure which is present over here. It will get activated, which is present on the inner side of the cell membrane, okay, or inner side of these receptors. This is something which is called as FAD. Okay, it is called as fast associated death domain. Fast associated death domain. As soon as these ligands or TNF alpha come and bind to these TNF receptors or fast receptors or CD95 receptors, which is nothing but the fast one, these fast associated death domain gets activated. These get activated. Now what it will do? It will link all these three receptors together, and this is something which is called as trimerization. This is something which is called as trimerization. Okay, and this trimerization will initiate the procaspase 8, or will convert the procaspase 8 to functional caspase 8 activation. Caspase 8 activation. This procaspase 8 is normally present in the cytoplasm. It is non-functional. To make it functional, trimerization of these receptors, okay, will initiate this procaspase 8, and will make it activated. So we have the activated caspase 8, okay, and ultimately this activation of caspase 8 will further lead to increase activation of caspases 3, 6 and 7, which will again lead to increased activation of proteases and endonucleases. And ultimately, if these enzymes are activated, it will lead to cell death. It will lead to cell death. So fast ligand or TNF alpha, basically they are released in severe damage, okay, and they come and bind to these receptors, okay. They come and bind to these receptors. What will happen? Trimerization will happen. Trimerization will basically, it is the fast associated death domain will activate, which will in turn make this procaspase 8 turn into active caspase 8, which will in turn activate caspase 3, 6 and 7, which will in turn activate proteases and nucleases, and ultimately if the proteins and the DNAs of the cells are destroyed, what will happen? The cell will die. The cell will die. So just give me a thumbs up, guys, that you all understood this. Before that, just answer me, what is the initiator caspase in this particular pathway? Now, two questions that you will answer me. If we are talking about extrinsic pathway, extrinsic pathway. Siddhi, perfect. But just answer me. What is the initiator caspase? Yes, initiator caspase over here. And the next one, that is the executioner caspase over here. Executioner caspase over here. Yes, Sakshi Kumar, you are very much right. It is the initiator caspase is 8. Why I'm writing like this? Because E starts and it is the extrinsic pathway. Extrinsic में E और intrinsic में 9 याद रखना है. Executioner, it is same. It is 3, 6 and 7. Yes, Osama, right. Prikshit, right, beta. Perfect. Perfectly right. Very good. Very good. I hope the mechanism of the extrinsic pathway, okay, and the intrinsic pathway is clear. The pathways of the apoptosis is clear. Just give me a thumbs up in the chat box, guys, so that I can start with the next topic, that is the silent features of apoptosis. Yeah, just give me a thumbs up, guys, so that I can start with the silent features of apoptosis. Yes, Siddhi Kakkar, very good. Osama Sheikh, very good, very good, very good, guys. So yes, I hope you all have done your writing along with me. All the tables, all the pictures are clear to each one of you. Yes. Prikshit, Ayman, Ahmed, very good, beta. Chal. Now we'll start with the silent features of apoptosis. Just give me a 1 minute break to each and everyone. Just a 1 or 2 minutes break to each and everyone. So that we'll start with the silent features of apoptosis. Sure. Let's start. Buckle up your seat belts, guys. I mean, the very first one that I have discussed in the table, it is about the cell size. Now, from the table, guys, just tell me what happens to the cell size in case of apoptosis. What happens to the cell size in case of apoptosis? What happens to the cell size in case of apoptosis? Yes, Sakshi Kumari, very good, beta. It decreases. The cell size decreases. And it is automatically referred to as the shrinkage. The shrinkage. The cell size decreases, and you will highlight this particular word, that the cell size decreases in case of apoptosis. The cell size decreases in case of apoptosis. Yes, everyone is right. Coming on to now why the cell size decreases, you already know it is due to structural proteins. Proteins are basically the building blocks of anything, okay, of the cells also. So if the structural proteins are destroyed, the cell will lose, the size of the cell will automatically shrink, no? So it is the structural protein destruction by proteases. By proteases. Due to structural protein destruction by proteases. I've already discussed. And remember that decrease in the cell size is one of the earliest changes. It is one of the earliest changes. It is one of the earliest changes. Now coming on to the next part that I have already discussed with you people, that the cell membrane the cell membrane, it is intact. There is no change in the cell membrane, guys. Why? Because there is no phospholipase activation. There is no phospholipase activation. Cell membrane, it is made up of phospholipids, and to destroy that phospholipids, we need an enzyme that is called as phospholipase. And during the whole process, we have seen the intrinsic and the extrinsic pathway. Phospholipase is not involved. No phospholipase activation, so the cell membrane will not be damaged. And if the cell membrane is not damaged, it clearly indicates that there is no inflammation. There is no inflammation. This question has been asked a number of times that we have to mention. Okay, there is no inflammation in case of apoptosis. Okay, there is no inflammation in case of apoptosis. Apart from that, coming on to the third point, the third feature of apoptosis, it is chromatin condensation. It is chromatin condensation. Chromatin condensation. Now, already know. Now, suppose this is a DNA. Okay, 5 prime to 3 prime, 3 prime to 5 prime, a double helix structure of the DNA. Now, if apoptosis is happening, how will the apoptosis happening to this particular DNA? I already told you there is an enzyme that is the chromatin condensation, means it is breaking at several points. It is due to endonuclease activation. It is due to endonuclease activation, and it will break this particular DNA into fragments. You already know. Okay, and ultimately, remember two questions over here that has been asked in the exam, that it is the chromatin condensation, it is the hallmark feature of apoptosis, and it is the most characteristic feature of apoptosis. So it is the hallmark feature of apoptosis, and it is the most characteristic feature of apoptosis. Parrot, doctor is so smart, is high IQ and smart man. तुम्हारी इंग्लिश मेरे से भी ज्यादा खराब है. चलो चलो. Anyways, guys, did you note this particular thing? Yeah. ये लिख लिया तुम लोगों ने? मैं स्मार्ट नजर आ रहा हूँ. I'm Chinese medical student. I need more learning from smart man. So anyways, guys, I hope you have written this. Just give me a thumbs up in the chat box. Coming on to the fourth characteristic feature, that is called as the gel electrophoresis. Gel electrophoresis. Now, remember that. Now, if we try to see these nucleic acids, the DNAs, okay, the nucleic acids, the DNAs, now these nucleic acids, the normal nucleic acids, basically they appear as a single band, When we talk about necrotic nucleic acids, basically they appear as smear pattern, smear pattern. And when we talk about apoptotic nucleic acids on gel electrophoresis, basically they will appear as step ladder pattern. Step ladder pattern. So you can see these step ladder, this step ladder. So these step ladders pattern is basically indicating the nucleic acids in gel electrophoresis. If they are showing you this type of step ladder pattern, it clearly indicates this is the apoptosis. And if it is just coming like a smear, it is necrosis. It is necrosis. So on gel electrophoresis, this question has been asked a multiple number of times. They have given these images. And they have just asked a question, which one of them is depicting apoptosis or necrosis? Which is necrosis, which is apoptosis? And this was just a very basic question that they have put on in exams multiple number of times. So yes, I hope you all have written this. So these were some general features of apoptosis, the silent features of apoptosis. So we have completed silent features of apoptosis. We have completed the pathways of apoptosis, both intrinsic and extrinsic, along with the diagrams and the pictorial representation, along with the flowchart. Apart from that, I have considered pathological and physiological apoptosis with a variety of examples. Differences between necrosis and apoptosis, concepts, the genes, and the gangrene. Till now, I hope everything is clear to everyone. Now I will start with the next topic, that is called as the pyroptosis. Just give me a thumbs up in the chat box, guys. Yes. Prikshit Siddhi, very good, guys. Prikshit Siddhi, what about rest? बाकी सब सो रहे हैं क्या? Osama, very good, guys. So now I will be starting with pyroptosis. I hope. Hmm. Parrot, if you have any questions, please refer to the Telegram group for your doubts. I will be addressing it over there. Right now, I am discussing something very important in the class. So, yes. Now, guys, what do you understand by the term pyroptosis? Pyro, basically, it is like related with the term that is called as pyrexia, which means fever, which means increased temperature. Pyroptosis. Now, basically, what is the definition? It is basically a cell death. It is basically a cell death that is associated with release of interleukin 1. Interleukin 1 has one function, that is, you already know, it is a fever inducing cytokine. It is a fever inducing cytokine. Now, guys, let us just discuss the pathogenesis of this in a very simple flowchart. Now, if there is any toxin that enters the infected cell, okay, or if there is any microbial infection, okay, microbial infection or any infection, okay. Now, what happens? These are basically recognized. These are recognized by the intracellular responses. Responses बोल रहा हूँ. Yeah, it is the receptors, guys. The flow. Yeah, recognized by the intracellular receptors. Okay, and especially with your physiology knowledge, you would know we have node-like receptors, and these receptors are recognized. Okay, they recognize this particular cells. Okay, now what happens after that? There is a production of a structure that is called as inflammasome. 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 Okay. Now this inflammasome is basically leading to activation of two caspase, caspase 1 and caspase 11. Very, very important from the exam point of view. Very, very important from the exam point of view. It is leading to the activation of two different caspase, that is caspase 1 and caspase 11. Caspase 1 further leads to interleukin 1 activation, which will lead to nothing that is called as fever, that is called as pyrexia, or that is simply the term which is called as pyroptosis. Pyroptosis. Apart from that, guys, remember we have caspase 11. Caspase 11 will lead to cell death. Caspase 11 will lead to cell death. Caspase 11 will lead to cell death. Okay. So remember, two questions are being asked over here. Okay. My audio is visible to each and everyone. Okay. Yeah, inflammasome, hercine, sing. Basically, these are okay. These are actually the receptors or the sensors which are actually regulating the activation of these caspases, caspase 1 and caspase 11. Okay, basically in response to the various microbial infections or toxins, these inflammasomes are basically the sensors which are actually regulating this. Okay, they have been derived from the host proteins. If you are talking about the morphological structure, they have been derived from the host proteins. Okay, so yeah, infectious microbes and उसके protein से inflammasome का structure बनता है, if morphologically talking about that. Okay, apart from that, guys, remember we have two morphological features that you have to remember. Morphological features, that is cellular swelling. Cellular swelling is present over here, and apart from that you will encounter that is something called as inflammation. That is something called as inflammation. Cellular swelling and inflammation will be present in case of pyroptosis. Yeah, I hope this is done. I hope this is done to each and everyone. Just give me a thumbs up, guys, that you all have written this. Audio is not visible. I'll kill you, Kanchandeep. Okay. Is this done, guys? Just give me a thumbs up. Yes, Osama, perfect beta. What about rest? Pyroptosis is done. Yeah. Now coming on to the next part, that is the necroptosis. I hope everyone is listening me out over there. Yeah, I hope everyone is writing notes. Osama, Siddhi, perfect beta. What about rest? बाकी सब सो गए क्या? बिल्कुल ही चलो, perfect. Chalo. Coming on to the next part, that is the necroptosis. Basically, necroptosis is simply a hybrid. It is a hybrid of necrosis and apoptosis. It is a hybrid of necrosis and apoptosis. Okay. Now, what is actually happening in necroptosis. Basically, mechanism is of apoptosis, but morphological features. दीपेश सिस्टमिक पढ़ाए सर एग्जाम इज नियर प्लीज ये टेलीग्राम ग्रुप पे लिखना कि यू नीड क्लासेस ऑन अ रेगुलर बेसिस तो विल प्लान इट अकॉर्डिंगली या कमेंट सेक्शन में आफ्टर द क्लास morphological features are of necrosis. It is also known as programmed necrosis, and one more thing, that it is caspase independent. It is caspase independent. It does not require caspase. It does not require caspase. It does not require caspase to function. Now guys, listen. Now guys, listen, that it is seen in physiological examples. If we talk about, we have, it is seen physiologically also, physiological and pathological as well. Pathological as well. Physiological, basically it is involved in the formation of mammalian growth plate, and pathological, it is involved in acute pancreatitis, neurodegenerative disorders, also seen in inflammations of the liver, the gallbladders, so we can say hepatitis, okay, so there are multiple conditions also, okay, Parkinson's also. So yeah, these are certain examples of physiological and pathological basically the cell starts at apoptosis but it ends at necrosis. Basically the mechanism is of apoptosis but the features are of necrosis. Why features are of necrosis? Because there is no caspase involvement. It is caspase independent. There is no caspase involvement death. No caspase involvement. So we'll just try to figure out the mechanism right now. We'll just try to figure out the mechanism. So you already know that. Just try to draw the structure along with me. Just try to draw the structure along with me. You already know we have these receptors. Okay, these receptors which are present over here. Okay, and you know this is the cell membrane. This is my dear cell membrane, and these cell membrane are containing certain receptors. You already know these are TNF receptors. Okay, these are TNF receptors. Okay, TNF receptors, and over here the TNF alpha is coming and binding along with the fast ligands. Okay, we have fast receptors or commonly known as the CD95. Fast or CD95, it is the same. So this is something which is you guys have discussed. Now what is happening over here? Now over here we have FAD also, but there is no involvement of FAD over here. So what is happening? The TNF, okay, comes and bind with the TNF receptor directly, and it will lead to the activation of RIP, okay, that is called as receptor associated kinase 1 and 3. Now we doesn't have any FAR involved over here. Now don't confuse it with the extrinsic pathway. It is simply the TNF has come and combined with the TNF receptor. And what is happening, guys, over here? There is activation of receptor-like or associated kinase. RIP stands for receptor associated kinase. Okay, and that is 1 and 3. Now this receptor associated kinase um, will activate the reactive oxygen species. Okay, that is the reactive oxygen species. And this receptor associated kinase will not lead to no लिख लेना बड़ा बड़ा no caspase activation. No caspase activation. No caspase activation. So reactive oxygen species. Okay, now this reactive oxygen species will lead to mitochondrial damage. Mitochondrial damage. Now if mitochondria is damaged, you know the other structures such as lysosomal membrane damage. And one more thing, that is inflammation. If cell membrane is damaged, inflammation will surely be present over here. And apart from that, you will see a decrease in amounts of the ATP because mitochondrial damage is involved. The mitochondrial damage is involved. So that is all about the necroptosis. Pyroptosis I have already discussed. Necroptosis we have discussed the examples along with the mechanism. I hope it is clear to each and every one. Okay. Now coming on to the last one, that is the ferroptosis. Okay. Coming on to the last one, that is the ferroptosis. Now, now this pathway, as the name suggests, it is involving iron. Okay. Now this pathway can be regulated by reducing the iron levels. By reducing the iron levels. Okay. It plays a role in cancer and neurodegenerative disorders. Neurodegenerative disorders. Okay. Now, what is the mechanism, guys? What is the mechanism? Remember that if there is excess intracellular iron, now suppose if there is excess intracellular iron, that is Fe, what will happen? It will saturate the glutathione antioxidant pathway. Antioxidant pathway, which will lead to excess free radical damage. Free radical damage. Okay, which will again lead to unchecked lipid peroxidation of the membrane. Lipid peroxidation of the membrane. And if the lipids are destroyed of the membrane, what it will in turn do, the plasma membrane will damage. The plasma membrane damages, or plasma membrane is damaged, And it is something which is, when the plasma membrane is damaged, it is death like necrosis, because in necrosis only you will encounter the plasma membrane is damaged. Necrosis only you will encounter the plasma membrane is damaged. Death like necrosis. Ferroptosis is done. Necroptosis, pyroptosis, all done. Yes. So just give me a thumbs up, guys, that you all have like done, understood all the concepts with me right now. All the concepts, everything is done. Yeah, just give me a thumbs up, guys, that all the concepts, everything is done. I'm waiting for your responses, guys. I hope till now everyone like wrote all the notes and understood everything. Guys, yes, Prikshit, perfect beta. What about rest? All attentive in my lesson? Like, Sakshi, Prikshit, perfect beta, perfect, perfect. It's somewhere lagging or my internet is not working. Sure. Coming on to the last one for today, that is cell pigmentation. That is cell pigmentation. I'll not be discussing this in like detail, because I'll be discussing it in more subsequent topics as well, where I will be talking about the stains and everything also. But for the time being, just remember the very first one, that is the melanin. Sakshi, Ayman, perfect beta, perfect, perfect. First one, that is the melanin. Okay, melanin, you already know melanin is responsible for your skin color. Okay, it is the endogenous black pigment. Endogenous means which is synthesized in our body, which is synthesized in our body. Okay, endogenous black pigment. Okay, now what is the function of this melanin? It provides hair and skin color. It provides hair and skin color. And remember, according to your biochemistry knowledge, that derived from it is derived from tyrosine. It is derived from tyrosine. Apart from that, the another cell pigmentation that you should remember, it is hemosiderin. Hemosiderin. Remember, hemo means, you already know, coming from the word iron, or it is derived from iron. Okay. Now it is present at storage sites of iron. Where iron is found is actually the bone marrow. It is actually the bone marrow. Okay. And hemosiderin, basically, it is detected by Prussian blue. It is detected by Prussian blue. Okay. And you already know these Prussian blues are purple or violet granules. Okay. These Prussian blues are purple or violet granules. Okay. And the reaction is known as Pearl's reaction. The reaction is known as Pearl's reaction. I'll be discussing it in inflammation also, don't worry about this. Apart from that, the third one, that is the lipofuscin. Lipofuscin, okay, or it is the lipochrome, okay. You can see around the nucleus, you can see the brown color dotted pigments around the nucleus, you can very well imagine. So this is a perinuclear, means around the nucleus. It is a perinuclear brown pigment. Perinuclear brown pigment, okay. It is a lipid derived pigment, okay. Lipid derived pigment, okay. It is accumulated by lipid peroxidation. And remember that lipid peroxidation, it is associated with free radical injury. It is associated with free radical injury. It is associated with free radical injury, okay. So it is seen in aging, okay, protein energy malnutrition, cancer, okay. In aging, we also call it as wear and tear pigment, okay. Wear and tear pigment, it is associated with old age, brown atrophy. It is classically called as brown atrophy because around the nucleus you will see these brown pigments, okay. And coming on to the last one, that is the calcification. And already we know the calcification, it is of two types. Dystrophic calcification and metastatic calcification. Now in dystrophic, remember the serum calcium is normal. Whereas when we talk about metastatic calcification, guys, the serum calcium, it is increased. Remember, dystrophic, the word D says it is deposited in dead and degenerated tissues, okay. And over here, the metastatic one, it is deposited in living tissues. It is deposited in living tissues. And yes, remember the examples of dystrophic calcification. The very first one, remember, it is the TB of the lymph nodes. TB of the lungs. The second one, it is the cardiac valves, where you can see dystrophic calcification in rheumatic heart disease. Atherosclerosis, and the tumors, cancers, where you can classically see schwannoma bodies. You can see it over here, schwannoma bodies. These are onion को जब बीच में से काटते हो, slices करते हो, you just see the onion shaped balls over here like this. These are schwannoma bodies, okay. Schwannoma bodies, okay. Schwannoma bodies. Apart from that, guys, remember tumors कौन से? You will remember it by the mnemonic most. It is meningioma. It is the papillary cancer of the ovary, papillary cancer of ovary. Okay, it is seen in papillary cancer of salivary glands, and it is also seen in papillary cancer of thyroid. It is also seen in papillary cancer of thyroids. Apart from that, if we talk about the examples of metastatic calcification, yeah. If we talk about the cancer of the parathyroid gland, that is parathyroid adenoma, we will discuss it later. Okay, you will see the metastatic calcification. Okay, apart from that, you can see it in vitamin D intoxication, vitamin D intoxication, and you can also see it in milk alkali syndrome. Milk alkali syndrome. So that's all for today. The calcification. I have discussed the different examples, the two types, dystrophic and metastatic. I have discussed the 2-3 types of cell pigmentation. I have discussed ferroptosis, necroptosis, pyroptosis. I have discussed the silent features of apoptosis, the extrinsic pathway, intrinsic pathway, mechanism, action, the pathological apoptosis, physiological apoptosis, differences between necrosis and apoptosis, the genes, and which genes are important, I've all mentioned in the table over here. I've discussed the gangrenes and the types of gangrenes with pictures. I hope today's lecture is clear to each and every one. Today I'm just ending up the cell injury. Okay, so I hope everything is clear to each and every one. So please share the video, share with your friends. They can also benefit from it. Okay, guys. I hope you all understood. Please like, comment in the comment section afterwards when I just shut. Okay, and keep working hard, beta. Keep working hard. Yes, keep working hard.